So hello everybody and welcome to this Pint of Science event today. It's the Pint of Science edition from 2021 today. And my name is Juditha Schmidt and I will be your host for this evening in which we hear about fascinating science. So Pint of Science already presented two exciting evenings in the previous days with the titles Identification for All and The Waves of Radiation. So if you couldn't attend these events, you can still find the streams on our YouTube channel and watch them today or in the next days. Today is day 3 of our event and our topic is A Day in Science. We will hear about how scientists work, may it be in a remote research station, in a lab, or with animals. So we are looking forward to these 3 very interesting insights into research. And of course, this would have not been possible with the help of lots of engaged volunteers here in Oslo, taking care of speakers, technical aspects, sponsors, and of course as well coordinating this event. So thanks to everybody who made it possible that we can enjoy this evening today. And of course we want to thank as well all our sponsors. We are very happy that so many different institutions and organizations support Pint of Science. Our sponsors are UiO Doc, the Norsk Biokemisk Selskap, Committees Oslo, Mona Lisa Bites, the Nansen Legacy, Simular, and the Center for Digital Life Norway. And another one of our sponsors is the Akademie for Yngre Forskere. And today, one of our speakers, Helene, is representing them and she will say a few words about them now. So therefore I give the word to Helene, who will introduce us now to the Akademie.
Yes, so the Young Academy of Norway is a diverse, multidisciplinary and cross-sectoral meeting place and a research policy platform for younger researchers. So it's also a driving force for innovative research dissemination, an attractive arena for scientific debate, and a significant and also visible participant in research policy issues. So currently we consist of 44 members based all over Norway, and we are now also looking for new members, and it is possible to apply for a membership at the moment. So that's it.
Thank you, Helene, and for this nice introduction to the Akademie. And now I want to introduce you to everyone who is involved in this evening. So I would like to invite all the participants here on the stream. So I think that's all perfect. So our first speaker will be Coline Bouchayer from the University of Oslo, and her title for today is A Year on Ice. So hello, Coline. And our second speaker will be Helene Knævelsrud from the Institute for Cancer Research at the Radium Hospital that you have just heard. And she will talk about fruit fly sex in cancer research. So welcome, Helene. And our third speaker for today will be Abril Mckay from the Norwegian University of Life Science. And the title of her presentation today is A Year in the Life of Bats in Oslo. So hi, Abril. Nice that you're here as well. And of course, besides our 3, fantastic speakers, more volunteers will take you through this evening today. So Lavinia and Praveen will take care about all technical aspects for us. So thanks a lot for that, for being so active in the background of this stream. And Elisa has worked as well in cancer research in the last 10 years, and recently she moved to the biotech sector, and she will be managing the questions for today. And last but not least, Lourdes is doing her PhD at the University of Oslo about archaeogenomics and marine biology. And she will take care of the quiz, and she will tell you a few words about this before we start with the presentation. So please, Lou.
Hello, everybody, and welcome. Just a few quick words about the quiz for tonight. So we'll be playing in Kahoot. Maybe you all have heard of the app. So if possible, please download Kahoot in your phones. But know that you can also play in the computer. So for that, you just go to www.kahoot.it. I will repeat this later on, no worries. And also, please, when connecting to the game, use your name as your nickname, also to be easier to identify the winner later on. And yes, we need to identify a winner because there's going to be a lovely prize for the winner. The prize is a surprise, so I cannot really reveal anything else. But, well, that's all for now, and just stick around and enjoy the event.
Thank you, Lou. And I think now it's time to start with our first talk of tonight. So please just leave now.
Yeah, so I just need to share my screen.
Yeah, we'll give a short introduction to you first. So this is Coline that you can see now. She's a PhD at the Department of Geosciences at the University of Oslo. And the focus of her work is glacier friction. So she studies how a glacier moves over the underlying ground. And to do so, she investigates glaciers on Svalbard, and she just come back from the high Arctic 2 weeks ago, so we are very happy to see her here today. But today she will not take us to the north, but to the very south, to a research station in Antarctica. So now I give the word to Coline and enjoy her talk.
Yeah, thank you. But apparently no one can see my screen. Okay, good. Yeah, so before digging a little bit on really the subject, I wanted to give you a short appetizer on what I will talk about today. And since cinemas are closed, I thought that a little trailer will be a good thing. So enjoy watching, and I come back just after. Good. So that was a little appetizer of my talk about today, which is called The Year on Ice. So I made it as a poster or cinema presentation. So basically, I was living in a station in Antarctica for an entire year without possibility neither to come back, neither to get rescued or whatever. And so my talk today is about getting a little bit the feeling of living in such an environment for a year. So first, I want to bring you from a little journey, like to Norway, to this white, extreme, and the driest continent on Earth, Antarctica. So this continent that's so this is going again, yeah. This continent that we don't necessarily get the size of, because usually because of the projection, this is super small at the bottom of the map. But this is actually a very big continent. So here you have a map with the comparison of the size of Europe. And so you can see that this is really, really big. Since even including Russia, this is not even covering the entire Antarctica. But there is some people that live there for a year or less than a year, depending. And so here you can see all the research station in Antarctica. And Antarctica is a very special continent because it doesn't belong to anyone. And this is a country or a continent for peace and science, as it described by the Antarctic Treaty that protects Antarctica. So I was in Concordia Station, which you can see it's with this red cycle around, which is one of the 3 stations that sits on top of this huge ice cap. It's really, really remote, so we are 1,200 kilometers far from the coast. So yeah, quite cold and remote extreme environment. So technically, how to get there? So it's a little bit like every job. Like you get first an interview, but what changed is that, as I tell at the beginning, this is quite impossible to get rescued. So we need to go through some medical check to be sure that all our body is fine, as well as some psychological tests to be sure that we can sort of handle in the best way this extreme environment. So this is basically coming from the world, or this picture shows the world maybe without the pandemic, but the world that it used to be, to a reduced society in summer. And in winter, then we are going to a macro world. What you need to imagine is that summer is the opposite way, because this is the southern hemisphere. So when it's winter here, this is summer for us. A very cold summer still, but summer. And then from February on till the next October, this is what we call winter. And so in winter in Concordia, there is only 13 people that stay there to maintain the entire station. So this station is quite unique because it's run by the French and the Italian. So this is a cooperation, and this is the only station which is run by 2 countries in the same time. And so in summer, there is approximately 80 people and 13 people only in winter. The maximum temperature, so our tropical summer there is minus 35 degrees, and minimum temperature in winter are minus 80 degrees. That makes an average of approximately minus 55 degrees. To give you an idea, your freezer is approximately at minus 18 degrees, so it's way colder. And if it was not difficult enough, then Concordia is as well in altitude, standing on top of 3 kilometers of ice. So we are at 3,200 meters above sea level. So this is the station where I spent 1 year with my colleague there. And basically how it's organized, there is 2 towers. There is 1 tower, which is called the Calm Tower, where we have the hospital, we have our room, and on the last level, we have the labs and the radio station. In the other tower, this is the noisy tower, so more where we live. So on the bottom of it, there is the workshop for the technicians, where they're working with the electric power station. And on the second level, we have a sort of cinema room, sport room, and all the magazine for the cook, so where there is all the food. And then on the third level, this is the kitchen and the restaurant, as well as the living room. So really the living area. So who is needed there? Actually, everyone, because we are a micro society, but we need to provide everything for an entire year. So we have a technical team that get in charge of, well, producing electricity and as well heat the station, which is quite important with this temperature, as well as producing water. So producing water there is basically melting the snow, and then we recycle 80% of the water that we are using. And as well, driving engine to remove the snow that is blown by the wind, and repair, of course, this engine, because none of these engines are really made at the beginning to stand this really cold temperature. We have people that take care of the scientists. So we have a cook, which is really nice. That was an Italian cook, so we ate a lot of risotto and pasta. And then there is the hospital, because since this is impossible for a plane to land in winter because the fuel might freeze, then we need to have a doctor in case of emergency. Luckily, almost nothing happened, and no big injuries have never been reported, at least in Concordia. And then, as I tell, there is this radio station, because whenever we are out, we need to warn people, yeah, I'm out, I'm going here, I will come back at this time. As well as in summer, they serve as an airport station. We are making land, we are helping the plane to land and in which direction, and communicating with the pilots and so. But as I tell, Concordia, or Antarctica in general, is a continent for science. So there is a lot of science going on there. So here are some pictures you can see on the left-hand side with these two big green laser. This is some lidars, and they are making some atmospheric composition measurement to better understand the climate. Then on the bottom, you have an instrument that measured the magnetic field of the Earth. And on the top, you have a flying simulator, because there is some human space research there. So we were all involved into human experiments. And so we were trained in summer to fly a simulator that is the same or a little bit simplified simulator that is used to land on the ISS. And so the Carmen that you can see here was the doctor that analyzed the data and see how our performance on landing on the ISS evolve through all the winter. Then on the left again, of course, we have 3 months of polar night. So that makes Concordia a great place to study astronomy. Then you can see on the left, so the picture with the southern aurora. There is some investigation about space weather. So basically how sun eruption and so on is interfering with the envelope of Earth. So that makes aurora, but this is as well really interesting to study certain behavior. On the top, you can see Filippo, who was a meteorologist, and he's now blowing a balloon. And this balloon was launched every day and takes some measurement about the wind, the pressure, temperature through all the atmospheric column. And on the picture down, you can see the group of, we were 2 glaciologists studying snow processes as well as ice processes. And here we are standing in a snow pit to collect snow samples. But we are scientists, but as well there we need to be rescuers and firefighters. And so we were trained through all the year to, in case of emergency, so wherever someone, for example, fall in the stairs outside, how we'd rescue them, because of course you can imagine that minus 80 degrees, you don't survive really long outside if you're just injured somewhere and cannot move anymore. But the biggest danger is fire, because if the station gets on fire and no one can rescue us, well, we just don't have any house to live anymore. That is quite problematic. But hopefully we have some free time. So people tend to think that in Antarctica we are bored and we need to find a lot of books to read or someone to really annoy throughout the year because we just have nothing to do. But actually we are 13 that are always together. So it's like living in a house with roommates, but in 13, and we didn't choose the people that we were with. That makes it sometimes challenging but exciting in the same time. And as well, there is a huge minority of girls. So we were 2 girls out of 11 guys. And I was the youngest when I was there. I was 23, while the oldest was 56. So there is as well a big gap in age. And so people comes with very different life experience. But we feel greatly our free time, so we cooked sometimes, we made parties. So you can see, for example, on the picture on the top right, that we were a little bit missing some sun. It was in July, and we were just missing the warmth. So we just put the heaters up we project the sea, and we were just enjoying being in a baking suit inside, but that was better than nothing. We had a sauna, then we made a little bit like Pint of Science right now, like some popular talk to explain to the technicians as well what we are doing. We built the climbing wall, and we made as well the picture on the bottom, the walk for climate. And this picture then came through a lot of media because that was quite a symbol to make a walk for climate in Antarctica. So I missed a D in this slide. So to conclude, this is really an incredible experience that I don't regret to do. And to be honest, if someone would have told me 10 years ago that I would be one of these 13 scientists that will go in Antarctica and live this extreme experience, I would not have trusted them. And so my main message to like everyone, and as well like tell children and so on, that there is no dreams that are impossible as long as you believe them. And you just need to create the opportunity to realize your dreams. So for me, Antarctica was my dream, and I managed to go there, and that was just crazy. And we need everyone in Antarctica. So there is no more intelligent, less intelligent, or whatever. We need everyone. So the quote from Walt Disney illustrates, I think, well this, that a dream can come true if you have the courage to pursue them. So yeah, that was all from my side. I don't really know how much was the time and so on, but I thank you all the Pint of Science organizers to give me this opportunity to present a little bit of Antarctica, and I'm pleased to answer the questions about it. Thank you.
Hi, thank you.
Hi.
Thank you very much. This was so cool. I'm Elisa, and I will be managing the questions. So I think this was a really interesting story. So it's I'm a laboratory scientist, and for me, science is all in the lab. When you tell me minus 80 degrees, for me means only the freezer that we have. But I think this was super cool. And so I'm not sure if there are questions already, but I have a question. So you were selected to be one of the people that went there, and I was wondering if you tell me something more about what kind of training that did you have to undergo to be prepared enough for this experience.
So there is no specific training, actually. So we are mainly trained in summer when we arrive here for all these rescue and firefighters problems. So like in summer, when there is more people and more experienced people, then we are trained like this. But after, beside the medical check and the psychological, like it's basically questionnaires that they give you and that you need to answer. And as well, you have a 3 hours interview with a psychologist. But there is no like special training. We had like just 1 week of team building where we were in France with the Italian, going in some kind of island close to France, and that we would do like kayaking as well as some games, as well as some, like a team building for a company. So there is no specific training.
Okay, yeah, that's I guess it's a very psychological preparation that you need. So, okay, we have a question. Would you go back to Antarctica for a year if there was an opportunity again? He's asking you.
Yeah, no, I will not. I don't regret at all having lived this experience. I thought it was great. And if I came back in time and this opportunity would have come again, I would have definitely say yes. But a year is long, and you pass through the entire cycle. A lot of people come back, like so in the crew, which is this year, one of one of my friends that was with me during my winter over is actually again in Antarctica now. So some people really fall in love with this experience. I have lived it once. I know how it is. It went really well. That is not the case for all the winter hour. And so I have ticked this box. So I leave the place for someone else now.
Okay, Fiorito is asking if you miss Antarctica. Well,
yeah, I think I miss Antarctica. That's like this is like crazy souvenirs, and this is weird as well to think that this is a place that you have lived an entire year, that you will probably never come back again. So there is some really strong feelings that I'm sharing with my friends that I live this experience with, that we are the only one that can understand this kind of feelings, or I guess like not the only one, but for our experience at least. And so yeah, I miss this part. And as well, since it's a station run by French and Italian, then, and I'm living in Oslo now, so like it's not that I'm seeing them all the time. And it's definitely a society which is different. There is no questions of money, there is nothing like this. So yeah.
So yeah, so how do you manage waste disposal in the station, is asking Maria.
So we sort everything. So we have a lot of different plastic. So all the plastics are different. So you need to be careful in which stuff you put what. Then the food, we had a sort of big, I don't know the name in English, but a big, sort of container where you have bacteria that eats, the food waste. And then cardboard and glasses and so on, and everything, it's maintained by the technicians. So compacted, put in box that are put in container. And then when summer came, there is what is called the traverse. So it goes from the coast to Concordia, and this is a bunch of big tractors, or winter tractors, or same stuff that are grooming the slope in Oslo. And they are traveling to Concordia for 12 days, and they are in charge of taking all the containers of our waste to bring it back to the coast. Then the containers are put into the boat, and this boat is going back to New Zealand or Australia, and the waste are taken care of there.
That's really interesting. So actually, something I also wanted to ask you that Francesco is asking is what kind of research is conducted there, and what did you research on?
Yeah, so I was probably a bit quick in these slides of like science. So all that I presented about like meteorology, glaciology, space weather, magnetism, seismology, and so on, all of these fields are studied in Concordia, and we are usually one person in charge for each field of science. So I'm a glaciologist, so I was taking care of snow and ice measurements. More snow than ice, because there is 120 meters of snow on top of the ice. So of course we are not accessing it really easily. So it's more for my job was taking samples mainly. So we are, let's say, operator of science. We are not researchers there. I had like 5 research programs that I was taking care of. And so all the research and analysis is taken care in labs in France or Italy or wherever. And we are basically the little hands and the eyes of all the researchers.
Yes. Like, it's really interesting. Okay, then Sachin is asking, how do you keep in touch with your supervisor and family back home? How did you have a good Wi-Fi connection? That's something
So-
essential.
So no, we don't have any personal internet connection, neither on our laptop, neither on our work computer. But we do have computers that send mails, which is like really nice. And after we could just had the little amount of Wi-Fi to have WhatsApp, but only message. So we couldn't call or stuff like this with our personal phone. After there is a satellite phone, so we could call with it, as well as one computer that had Skype. But you needed to be really patient to communicate. And so, yeah, to open your home in Facebook was probably half an hour, to give you an idea of how it's slow. So
Yeah.
Skype is not really well working.
I have a curiosity. So, from your research on snow, so could you see some kind of chemical? Okay, you didn't perform the analysis. You are sending your samples, but is it possible from composition and microbiological population to see some effects of climate change on that?
Well, okay, so first, the microbiological, there is nothing. So
Okay.
like we had a astrobiologist with us that was like doing other stuff, but he tried to put in place a project about this, and there is strictly nothing. After it's really far and really remote. So in Antarctica, there is some stations that can see the effects. Us, we are more working on the chemical cycles, so the general cycle, and as well as photo elements that react with the sun and so on, and as well how they sort of travel into the snow. So because of course they get deposited somehow, but when it's snow there, you cannot see that snow. This is this diamond dust how we call it. And so we are interested to see how this element that get deposited on the surface then travels through the fern.
Okay, thank you. Okay, we have one more question that is also something I wanted to ask you. So about animals in Antarctica. I wanted to ask if you saw penguins.
So there is a lot of animals in Antarctica, but all these animals need to eat somewhere. So basically, where they found where they can eat is on the sea. And so that's why there is like penguins and so on. So I've seen some on my way back from Antarctica. But in Dome Sea, we are at 1,200 kilometers far from the coast. So there is no animal, there is nothing, just because the people, like the animals, just cannot eat anything. It's so sterile environment that there was really nothing. Just us, if we can consider us animals.
Okay, thank you so much. I think I can now give back the line to Juditha for the next speaker.
Yeah, thank you.
Thank you.
I'm so sorry, I was muted. So our next speaker will be Helene, and I will ask her now to come into the stream and join me here. Thank you. So Helene is a project leader at the Oslo University Hospital, and as we saw earlier, a member of the Akademi for Yngre Forskere. And she has a great passion for cell biology, and in particular for the study of the mechanisms driving cancer. And during the last years, Helene has performed her cancer research in an organism which is very tiny but can model human diseases well, as I've heard, the fruit fly. And today, She will give us some insights into her work, and I maybe add, like, if you have any questions, you can already post them in the chat during the talk. So thanks, Helene, for giving a presentation today, and please go ahead.
Thank you. Yes. So what I will present to you today is about fruit fly sex in cancer research, because as was presented, I'm a project leader at the Institute for Cancer Research at Oslo University Hospital. And what we are wondering about as cancer researchers is, how does a normal cell transform into a cancer cell? So this is one of the questions that we want to address in the lab. And we also want to prevent this development of cancer and get rid of the cancer cells. So one example is our bone marrow. So in the normal bone marrow, during normal conditions, you have stem cells that will divide and give rise to all the different kinds of mature blood cells that we need. So the ones that transport oxygen, and the ones that fight infections, and the ones that heal wounds. However, if something goes wrong, then instead you'll have an accumulation of these immature blood cells. And these will disturb the function of the normal blood cells. And this is what is known as leukemia, or blood cancer. So to study this, we need to know a few things. So a human being each of us consists of many cells, and each of these cells will contain a nucleus with 23 pairs of chromosomes. So inherited one set from the mom and one set from the dad. And each of these chromosomes contain genes, and each gene provides a recipe for a function. Now, what we know is that in cancer, most of the time, one or several of these genes have a change, which will lead to a change in the recipe and a change in the function. This can then give rise to cancer, and often it's not only these changes or mutations, but also you need the function of other genes to support the development of cancer. So to study this, luckily, as researchers, we cannot genetically manipulate human beings or decide who will have kids with who. But when it comes to fruit flies, however, we can do exactly that. So fruit flies, they will mate, and the female will lay many eggs that will develop to larval stages. And eventually these larvae will crawl out of the food and pupariate, undergo metamorphosis. And out of this pupal case, there will come adult fly. And then the cycle can go once again. So for the research that I'm doing, I'm usually looking at this larval stage. And now you might be wondering, can fruit fly larvae get leukemia? And I would say yes. So I'll show you why. Here you're looking at the fruit fly larvae. So it's 3 of them to the left and 3 of them to the right. And what you can see in green is the blood system of these larvae. And the more intense green spot in each or area in each of the larvae is what is called the lymph gland, which is equivalent to our bone marrow. And what I have done to these fly larvae is that I have genetically manipulated one of the parents so that they express they contain a human gene variant that gives leukemia. And what you can see in these larvae is that this lymph gland, it becomes huge. So this is a sign that these larvae, or we take it as a sign that these larvae also show a leukemia-like state. So this we can use to figure out what other genes are important for this development of leukemia. But to do so, we need to mix then the genes that we want. And how we do this is basically to mate fruit flies. So what happens is that when a fruit fly female and a fruit fly male will meet, then first there is some flirting going on. He will touch her. She might kick him back, or she might accept him. And if so, then he will start singing for her by stretching out one wing and vibrating it. If this makes her convinced, then they go on to fruit fly foreplay, where he will lick her ovipositor. And when she's really ready, he will attempt to penetrate and eventually succeed. So at that moment, his semen will mix with her eggs and genes will mix. But for us, it's important to know that the larvae that come out of this cross are exactly the genetic composition that we want. We want to have full control of which genes they have. This means that the female cannot have already mated before. So one of the first things that we do every morning when we come to the fly lab is to look for virgin females. So here you see one example. We know that it's a virgin female because of this dark spot in the abdomen. So this is meconium leftovers of the larval tissue from the metamorphosis process. And this indicates to us that this female recently eclosed or escaped from the pupa. So every morning we collect these virgins to use in our crosses. And here you can see 5 young flies, and 1, 2, 3, 4 of them are actually virgins, whereas this one here is a male, which we can see by the genitalia. So you have to always make sure that the ones we pick are really virgins, otherwise all our crosses will be wrong. And here again you see 2 virgin females, 2 older females, and 2 males. So to then set up the crosses we want and mix the genes that we want for our experiments, we put together virgin females and males of a different genotype so that we can plan which genotype and which genes will be in the larval offspring. We also add some yeast, which you can see here, which basically works as an aphrodisiac for these flies. And for each combination that we want to test, we will then start one such cross. So you have the food down here, the yeast, all the flies, and then a closure to close off the vial. And then the fruit flies basically take care of the rest. So that makes it possible for me to reply to my question and figure out which genes support this leukemia phenotype. So here we have again the larvae with the leukemia-like phenotype. And if I have another cross where gene X has been removed in this larvae, you see that still the bone marrow like compartment, the lymph gland, is enlarged. And also, if I have the larvae from yet another cross, where still they express this human gene that gives rise to leukemia, and I have removed another gene, there is still no change. And like this, I will look through many such crosses until I find this one where gene Z has been removed. And you can see that the lymph gland is no longer enlarged. It's actually looking like the normal flies. So then I know that gene Z is involved in supporting the development of leukemia. And this is important knowledge for us, because now we can take this further in the lab and study this gene and the gene product in more detail. And we can either design or use an existing drug that targets this gene product, and then hopefully be able to use this to get rid of the leukemia cells. And with that, I would like to thank you all, and thanks a lot to the fruit flies.
Thank you so much, Helene.
You're welcome.
As a cancer researcher, I like this. It's very interesting, and it's incredible how much just fruit flies can do for research. I have some questions. So you said you described very detailed how how the genes can be propagated in and to generate a line of transgenic fruit fly. So I haven't worked with fruit flies myself, but I've worked with mice, and the first step to generate a transgenic mouse is to do microinjections. And I was wondering, the first step to create a fruit fly line, how does it happen? Like, is it an injection or how?
Yes, so that would be also an injection. So what you would start with is to microinject in fruit fly embryos. So you would inject a plasmid or a piece of DNA, and then you need to somehow select the ones that actually incorporate it into their genome. So usually this very standard strategy is based on eye color. So we can take advantage of certain features, external features of the adult flies. So the normal fruit fly that you find in your kitchen will have red eyes, but then it's a very common mutation that makes them have white eyes. And then in this white eye background, you can then inject in embryos that would give rise to flies with white eyes. And with your the DNA that you put in, this will give them a orange color eye. So then it's very easy to score that this and that fly actually incorporated this DNA into their genome. And then you propagate this line. So you have some tricks then to make sure that you don't lose it again. So you use something called balancer chromosome. So you know every time if the offspring inherited your gene of interest or another chromosome.
That's really cool. Okay, we have a question from Liliana. How long do flies live in lab conditions?
Yeah, so actually they can live quite long if you really take care of them. So I mean, when people do aging studies, then you can have the flies live for months if you really give them always fresh food and make sure that there's no infections and no contamination in the vials. But of course, usually we don't really need to keep them for that long. So for us, we might get rid of them or dispose of them after 10 to 20 days, maybe, depending on exactly what you want to do with them.
Yeah, I remember them flying around sometimes. Okay, so Maria is asking, is saying, first of all, that this is really interesting. And how many fruit flies do you need for this kind of experiments?
Yeah, so I mean, of course, I have then an incubator where I keep my fly lines. So I have then a collection of some 1,000 flies that have all each a different genetic background, right? And then depending on exactly how you want to look at this, so if you wanted to do a genome-wide search for all the genes that are important for leukemia development. So either you can then have specifically knock down one gene at a time. So then there's about 14,000 genes in the fly genome. So that means you would do 14,000 independent crosses, or even more if you wanted, several independent attempts at removing a gene, or you can try to remove kind of a larger area of a genome at once, and then you can narrow this down and then try to figure out which of the genes in that region is important afterwards.
Okay, we have a question from Coline, that is the previous speaker. So how long are these experiments?
Yeah, so we usually then start cross on, let's say, a Thursday, and then by the next week, 6 days later, like a Wednesday, you would actually have the larvae that you can look at. So then if you just want to look at the larvae or take a picture of the larvae, then in 5 to 6 days, I have the larval stage that I want. Then, of course, if you want to do more things like extract the DNA or extract protein and then follow up with other experiments, then you add maybe a few more days. But as soon as you have the different genotypes that you want to combine, if you can do one simple cross, then it's quite fast.
Okay, yeah. Yes, so you is asking a very interesting question that concerns ethical use of these animals. So does this research need to be approved by ethical committee beforehand?
So it does not need to be approved by an ethical committee. So this is different from then mouse research, for instance. So the flies are not considered as needing that ethical approval. However, we do have to work with them in facilities that have been approved. So since most of them, almost all our flies are genetically modified, we have to make sure that they are not released to the environment.
Okay, I have a question. So because, having worked with cancer, I heard of from you and from other people talking about cancer models in Drosophila, but I was wondering if there are other diseases that can be modeled and how close to humans can be.
Yeah, so fruit flies are used to model many different types of diseases. So they are quite famous for studies in aging and neurodegeneration. Also, more rare diseases are possible to model in fruit flies. So if you know that a certain mutation is found in a patient, but you don't really know what it does, you maybe just know which organ is affected. You can try to maybe make this mutation in the fly line and see if you have a phenotype there. And this can be quite informative. Then there's many different types of cancer, but of course that have been modeled as well. And a lot of this is then based on that human beings and flies share a lot of the same genes, right? So even though it's simpler in flies, but it's said that about 75% of disease-causing genes in humans, we actually find a fly version of it. So in that way, we are quite similar.
Yeah, it's incredible, but we are. And if we still have time, do we? I wanted to ask one more question about, so because you are representative of the Academy of Young Researchers, so maybe there might be in the audience somebody that could be interested in in this, and I wanted to know a bit how this academy has helped you in your research, or has been like a good experience for you to bring on your research with them.
Yeah, so I think that the academy is really a great opportunity to then meet other young researchers from all kinds of different fields. So it really is a possibility to get challenged and think broadly about research and research policy, and also about dissemination. So you also hear about very different topics and how people tell about the very complex scientific problems. So yeah, I would definitely recommend it if you're interested.
Okay, and yes, I got so now we about dissemination, and we were wondering if you are on Twitter or on some social media, so our audience can maybe follow you, and if you can promote there.
Yeah, sure. No, I am on Twitter, so you can search me up there. Yeah.
Okay. Okay. Thank you very much. I think we can now move on. Thanks again, Helene. And I can give back the line to Juditha. Screen is yours.
Thank you, and thanks, Helene, for this very nice and interesting talk. And now I would like to ask our third speaker to join me here on the stream. Yeah, hi, Abril. So this is Abril Mckay, and Abril is a PhD student at the Norwegian University of Life Sciences and a member of the Scant Bat Lab. And her research focuses on bat community dynamics in Norwegian production forests, but she began to work with bats much earlier in 2013. And there she worked with a variety of settings during that time. And as she says, she considers herself incredibly lucky to work closely with these animals. And as you will see tonight, Abril is constantly thrilled by the many mysteries out there waiting for the bat ecologists. So enjoy the talk of Abril. Please go ahead. Sorry, Abril, I think you are muted. Can you please unmute yourself so that we can hear you?
Oh gosh, that's embarrassing. Excuse me. Ugh. I was going to say
problem.
Thank you. Yeah, thank you. And thank you for the very nice introduction. My name is Abril, and I'm going to be giving you guys a talk today on a year in the life of bats in Oslo. You already heard a little bit about my background, but I'll cover it briefly here. Before I moved to Norway in 2018, I worked with bats mostly as an endangered species consultant in the Midwestern US. When I moved here, I started working with ScanBat during my master's, doing bat ecology research, and that's what I've been able to continue doing throughout my PhD. And I'm just in the first year of it now. So I want to give you a bit of an overview of what's going on with bats in Norway before I zone in on Oslo. This is a list of the 12 species that can be found in Norway. This is updated with the new 2021 red list. Of those 12 species, 8 of them are actually red listed or data deficient. And that's a problem considering the fact that they make up over 30% of Norway's mammalian species. The Norwegian government, Miljødirektoratet, they're aware of this issue, and bats are heavily protected in Norway. However, we have such large information gaps about where these bats are and when they are, that it's difficult to actually afford them those protections. So there's a lot of resources and energy being funneled into bat research so that we can develop bat management strategies and have better monitoring programs. But in the meantime, there's a lot of stuff we just don't know. To give you an idea of what that looks like, I'm going to pinpoint the Western barbastelle for a second. The Western barbastelle had been found in Norway back in 1896. A couple of researchers found, I think, maybe 1 or 2 individuals while they were out capturing bats. And that was the only record we had of the species in Norway up until 2008, when some members of the Norwegian Zoological Society found 1 individual in a mine in Larvik when they were doing a hibernacula survey. That's just a survey that NZF does around the Oslo Fjord region in southern Norway. They go around every winter and look for bats in mines and count the number of individuals of different species. But anyway, so they found this 1 barbastelle in this 1 mine in Larvik, and then since that occurrence in 2008, we find 1 and then sometimes 2 individuals in the same spot in the same mine every few years. Those are the barbastelles of Norway, these 1 to 2 individuals. We don't know what they're doing the rest of the year. We don't know where they are the rest of the year. And that is why they're critically endangered. That's not the case for all bats in Norway, but to give you an idea about where some of these knowledge gaps are and how big they can be for bats living in this part of the world. In Oslo, we actually have 9 species that we can consistently find throughout the year. This is data that I pulled off of Artsdatabanken today, and it's all observations of bats in the Oslo area. This is observations back into the 1800s. I probably could have chopped off some of those, but here it is. And just at a broad glance, you can see that bats are here all year. And it's a lot of it are observations of this 1 species, which is the northern bat. This one here. I've tried to color code everybody to make this easier as we go along. And then there's a sort of a nice spackling of the other 8 species, a lot of DeBenton's bats, and also quite a lot of the party-colored bat in the winter. We'll come back to that later. But here you get a sense for what the species are in Norway and how this fluctuates throughout the year for all the time that we have been recording bat observations. I'm going to start with the summer because that's where we are now, and because it's a really important time for bats. It's when the most bat species are active, and it's when bats are at their most active. In this part of the world, bats are having their maternity season right now. What that means is that females are getting pregnant and they're giving birth, they're lactating, they're raising pups. Those pups are learning how to become adult bats. And typically this is all taking place in maternity colonies. So bats within 1 species, sometimes a few different species will roost together in 1 place. This can be a house or a tree or some other kind of like anthropogenic structure or natural structure. Bats are very good at finding places to live. But in general, these maternity colonies involve a group of females and they're raising their pups together. Some species, like this brown long-eared bat, the males will roost with the females in those maternity colonies. They're not doing anything to contribute, but they're there. And then most of the time, males will actually go into their own little bachelor colonies. And in a few cases, we have some weird outliers here. This is unusual in the bat world, but we have 2 species that the males have massive bachelor colonies. Party-colored bats and DeBenton's bats can have like up to 200 males living together, which is really weird. Sometimes they're much larger than the females' maternity colonies. No one really knows what they're doing. Probably having parties and making a mess, but they're avoiding the females and the pups. So this is what the bats are doing in the summer in Oslo, and this is all the different species that are represented during these, May, June, July, August. And again, you see the northern bat makes up a big proportion of those observations. This bar up here is actually just chiroptera. So these are probably people seeing bats flying outside their balconies or outdoors somewhere and not being able to see them up close and identify them. And then down here, pipistrelles make up a large number of the observations. These guys are really common to be found near water. And if you're out using a bat detector where you can translate bat echolocation into a frequency where you can hear them, they have a very distinct echolocation. Their foraging calls are quite high, and it makes it sound like bubbles. So that's why people are probably picking those out a bit more. And they also will roost in bat boxes. So people who put up bat boxes will be finding them there if they're checking them. Good. So these are the bats in the summer in Oslo, and this is mostly what they're doing. Here you can see an individual bat roosting alone in a tree hollow. It's probably, I think that's a common noctule, yes. And down here, this is a pipistrelle bat that's roosting with its pups here in a maternity colony. These are squishy young bat pups. And this is what I imagine it looks like when you're learning how to fly as a juvenile bat, which is what is also a big part of the summer's bats learning how to be bats. Right. This is actually not a Norwegian bat, but I really wanted to give you guys an idea of how cute moms and bat pups can be, because that's a big part of the summer. Eastern red bats are from where I'm from the US, and they can have up to 4 bat pups, which is unusual. Most bats have 1 pup, and that's true for Norway as well. And you can see there's actually 3 with her. So there's 1 that's tangled over the side, going back that way, another going across her belly, this one's climbing on her head. She's handling this very gracefully. I just love this photo series, and it's hard to find really charming photos of bats, so I apologize if I've snuck in some North American species. This is mostly a talk about Oslo bats, I promise. So then we come to the autumn. In the autumn, at this point, the juveniles that were born in the summer are becoming very independent. They're quite helpless when they're first born, but they grow very fast. And in fact, most individuals, most males at least, are even reproductively active at the end of the summer. So they're getting kicked out of the maternity colonies. They're going to go off and live on their own, and they're trying to forage as much as possible before the weather gets cold. This is when breeding is going to start happening. So I mentioned that young males will be breeding, all the other males will be breeding. Females who are born in the summer rarely end up breeding in the autumn. It's a bit of a species by species basis there, but not all of the young females will be included. But essentially what most bat species in this area are doing is they're going out and finding each other in big clumps around known structures. This might be a hibernacula site, or near a hibernacula site, near an area where everyone's expected to go into hibernation later on in the late autumn and in the early winter. It might be a really tall tree for some species. They prefer to use those types of landmarks, or even a tall building. And they just fly in big flurries. And for lack of a better word, they have orgies. This is just as much condensed breeding as can be done, because what needs to happen next is most bats in this area need to migrate or hibernate or do some sort of combination, a hybrid of those two things. Bats do not handle the cold very well. They have very fast metabolisms. That's the cost of being able to fly as a mammal. And if there's no insects around to eat, then they can't afford to stay active in these types of colder climates. So bats are on the move with the seasons, with the season change here. The Pipistrellus natucii down here, you can see there's a band of observations in September. Oh, back up there a little bit. There's a band of observations here in September. That's because these guys are actually migrating along the coast. That's one of the only ways that we get observations of these bats, is acoustic recordings along the coastline in the autumn. So they are following the coast as they head down south. They will cross over the open ocean. They'll go between England and mainland Europe and cross all over Europe. They are big movers. And that's why they're popping up here. And then you see there's a big spike suddenly of party-colored bats coming into Oslo. So they're actually, what we suspect is that they're migrating here. They're not leaving, they're coming. And that's because we have these really nice tall buildings, which they like to use for their swarming. I'll talk a little bit more about that in the next slide. They have a very unique breeding behavior, and Oslo is very uniquely suited to it. So you've got migration going on, and then you've got bats trying to just eat as much food as possible to look good for breeding, I guess, but also just to prepare for hibernation, for turning their bodies down and finding a nice place to chill out for the winter. And I've pinpointed the species that are most common here. You've got the northern bat. Chances are that it's peaking in October and November because it's starting to find places to roost in people's homes, trying to find basements or other types of like man-made structures for hibernating in, not because of the party-colored bat, not the same reason as them. And then you see the pipistrelle here. There's also a lot of activity. I think that also has to do with more so breeding than anything else. All right. So the party-colored bat is interesting because they sing. Not many bats do this. It's a relatively rare phenomena. Pipistrelles do it as well, but not to the degree that party-colored bats do it, and not to the degree that they do it in Oslo. So the males, I'll back up for a moment. So this species was evolved essentially to roost in cliff faces along coasts. And that habitat has sort of rapidly been replaced, or at least subsidized, by tall buildings in cities. And also has a lot of that. So these guys will move into apartment complexes and find like the tallest open roof there, and a male will slice off that area as territory and start singing to attract females, and we assume also to defend that territory. But there's still a lot we don't know about how they're using this song. I've given you 2 examples of sonograms here. These are recordings that took in Oslo this last autumn. Up here you see echolocation calls of a bat that's just foraging or commuting. This is just a bat using sound to navigate space, nothing fancy whatsoever. And you can also see that the frequencies are mostly above 20 kilohertz. That is the end of the human range of hearing. That's ultrasonic. So you're not going to hear this, most likely, unless you have really, really good hearing. Your dog probably hears it. So this is a echolocating bat that's just trying to move around. It's an echolocating party-colored bat. And then down here, this much lower frequency call that's much more erratic and has waves and swishes. This is the song. And it's low enough that we can actually hear it, which is part of the reason that those observations spike, because people are watching these bats fly around the tops of their buildings by their balcony, and they're hearing them as well. Also, when they come in from migration, they tend to land in all kinds of weird places in Oslo. So people find them like right outside their front door and roosting on the sides of buildings, and that probably has something to do with it too. But anyways, I've actually got a recording of this call right here. So if you listen carefully, maybe you can hear it. But one more time, just in case. Now that probably doesn't sound very fancy to us, but the ladies love it. That is the song of the party-colored bat. So if you're walking around Oslo, especially near tall apartment buildings during the autumn, around October, November, and into December, you should listen for this because it's out there. Good. Oh, excuse me. So now we've come to the winter and the spring, because let's be honest, no one can tell the difference throughout most of these months here between December and April. During this time of the year, most of the bats that are in Oslo are hibernating, and they're going out to mines. There are quite a few mines around the Oslo fjord, and these are areas where the Norwegian Zoological Society are doing these surveys every winter, going into the mine with volunteers who are trained to identify bat species and to carefully record what they find. Yeah, so that's why there's a boatload of northern bats. It has a lot to do with those observations. And again, northern bats will use anthropogenic structures, so people who are not scientists are also encountering them and recording those observations. And bats will come out of hibernation for short periods just to get things moving, to get some water. They might even try to breed really quickly with their neighbor. And they might even be able to find some food. Bats don't technically actually hibernate. I'm not going to get into all the physiology of that because I'm not a physiologist, but because they have to get up somewhat frequently throughout the winter to move around and do these things, it's not full hibernation. It's actually just torpor, but we call it hibernating because it's easier. Right. So once it actually does become warm enough consistently enough and there's enough food and resources, then they're doing this emergence where they're leaving their hibernacula sites and moving toward breeding grounds and starting to get their bodies ready for either getting pregnant or, just getting ready for the summer in general. If you're a male, no one really knows what you're doing, but I'm sure you have to eat food. I should mention, maybe you've noticed here, that bats have delayed fertilization. So that means that they have copulation in the autumn during these swarming events. And so the sperm enters the female's bodies and then it just hangs out in there and it waits because no one wants to use that sort of like energy being pregnant when you're also going to be going through torpor and hibernation throughout the winter. So fertilization does not happen in the autumn. It's sperm is just stored in the female bat's body. And then once she emerges in the spring and she's actually fully active again, not just coming out for short bathroom breaks, then her body is like, okay, now we're ready. Now we're pregnant. So that's what's happening in the spring. Here you've got some photos of bats from a scientific article here that are roosting. This is a northern bat down here, and then this is a de Benton's bat on the left and a Brandt's bat on the right. These are both Myotis species, and they often roost near each other. And you can see that there's even condensation on their bodies. When they're in torpor for long periods, their bodies become the exact same temperature as their environment. They are basically just the wall, an extension of it. This is a photo of a bat from the US that is sick and has left hibernacula because they are ill. It's very unusual to see a bat flying quite like this, but bats will get out in the winter. And I wanted to show you what that might look like. Good. So in closing, I really hope I've managed my time well. Oof. In closing, bats are in Oslo. There's a lot of different reasons why they're going to be in Oslo throughout the year. But they're a really important member of our communities in Oslo, and they have all kinds of interesting roles to play. So I hope that you've learned to find a few different ways of identifying them. I hope that you can keep your eyes and ears peeled and notice them throughout the year now. So I'll open for questions if we have any time.
Thank you so much, Abril. This was really, really cool. And now I see also that they are around my place. So
Cool.
Yeah, I was actually wondering, how did you become passionate about bats?
Why did I start working with them?
Why did you, why do you like them so much?
Oh, okay. I don't know, like any good love story, it's complicated. I started working with them when I was 18. I knew that I wanted to work with wildlife from a really young age, and I had really cool parents who didn't know anything about working with wildlife, but they gave me all the resources I needed to learn and to find other people who could help me learn more. And so when I was 18, I had some experience working with wildlife, and that allowed me to get a job during the summer being a technician and assistant to bat ecologist. And I think it was really my second summer. I got put together with somebody from a different company than my first summer, and he was really, really enthusiastic about bats and about training me. And I think that helped a lot and helped me fall in love with bats, but it probably would have happened anyways. I can't imagine any other way. So I've been doing this my whole adult life. I'm a little bit brainwashed. It's difficult to answer. I'm sorry.
Do you have a favorite bat species?
Oh, I do. I think I have one for every day almost. I have a lot of them. The Eastern red bat that I snuck into the presentation is a personal favorite of mine. I also really like the tricolored bat in the US. In Norway, my favorite bat species is definitely the party-colored bat, the one that sings. Mm-hmm.
Yeah. So we have got some questions. So, okay, first of all, we have a comment from Olga that really likes your talk. And then I'm not trying to read this, I'm sorry. The name. So they're pretty gray. Why are they called party colored?
Right. So a lot of the photos that I showed you were of like the face, which is quite dark, like you're noticing here. But if you were to look at just the back of their hair, where all their actual hair is, they're very sleek and shiny, and it's sort of the same sort of coloration as a badger almost, but shinier. So they have like silver tips and a black dark coat, and that's where that name comes from. It's much easier to see in person.
So Maria is asking, she's saying it's really cool to get so many bat species in and around a city like Oslo. Do you get bat colonies in all larger cities in Europe and worldwide?
Yeah, that's a good question. Thank you for asking that. Urban bat populations are a thing. So in my first slide with the picture with the bridge, that's actually from Austin, Texas. Austin has massive bat populations because they've got these Mexican free-tailed bats, which love structures that people are making like bridges. So this Congress Bridge in Austin, Texas has one of the largest bat colonies in the world. and it's right down the road from the largest bat colony in the world, which is Bracken Cave. That's not in the city, but it's nearby. So for example, Mexican free-tailed bats in the southwestern US and in Mexico, they love cities, and they can get there in huge numbers. And in other parts of Europe, you'll find bats roosting in cities quite often as well. I can't shoot off as many statistics about it, but they're definitely there.
Yeah. Now that we know,
Yeah.
we will pay more attention to that. So Liliana is asking, so what is the ecological difference between the bats with big and pointy ears and the ones with small ears? That's really interesting, I think.
Okay, is it okay if I share my screen one more time so I can go back to the image? Okay, great. I'm gonna do that. Oh no, I think I was sharing my screen. I just stopped it. Why would I do that? Okay. So if I go back here, I think the bat that you're talking about are these brown long-eared bats, like this guy, right? So brown long-eared bats are ecologically, I mean, all species are ecologically different, but the advantage of having these really big ears is that they do have much stronger hearing, and the way that they utilize that is kind of cool. These guys are very quiet. They barely echolocate, and when they do, their calls are very, very low in volume. So they're sometimes called whispering bats. They take advantage of these really like large, powerful ears to be able to hear what's going on around them without necessarily having to echolocate as much. And there's even some examples of where moths are aware of this, and they recognize these sort of special quiet echolocation calls of these species, which tend to like those moths, and the moths will jam their sonar because they're so good at it that they have to find other sort of ecological tools for combating them. So that's one of the benefits of having really big ears, is that you don't necessarily have to echolocate as much, and you have a different sort of foraging style. But there's over 1,400 species of bats, and there's a lot of variety out there, so it's difficult to answer that in too much detail. I hope that helps.
Yeah, thanks. And what is a good place to see bats around Oslo?
Right, so actually Akerselva is very popular. This is from a report done by the Oslo Kommune on Nydalen. And they've pointed out that there's a lot of bats along the river, and it's true. If you want to go on a bat walk, I think it's easy to see them over the water. Even as it gets darker, it's sort of like a proper backlighting. And the Benton's bats, also known as the vannflagermus, the water bat in Norway, they will forage about like 3 centimeters off the water the whole night. They're just cruising along water surfaces. So it's really easy to go and find them foraging along freshwater ponds, but also rivers. And then I guess it a little bit depends on what kind of places you like to go. I think you can find bats in all places of Oslo. It's about knowing how to look for them. But this, like, the river walk is a nice thing to do, and it's pretty easy to spot them there.
One more thing to do when you stay in the city.
Yeah.
Okay, so Sachin is asking, during torpor, how much do they reduce their body temperature from their normal temperature?
That's a great question. I'm not a physiologist, so I don't have really specific figures on this, but I can tell you that it varies between species, and some species are better suited to coming out of torpor more quickly, and they have sort of ecological reasons for doing that, and it means that maybe they don't get as cold for as long. That's an example of why there might be differences there. But I do know that bats can basically get to the same temperature as their environment during torpor. So it just sort of depends on where they are and what species are there, and even, you know, the regions as well.
Okay, so Francesco is asking if, oh yes, this was something I also thought about. If you have any comments about the bad publicity that bats have accumulated during the last year as a virus reservoir during the pandemic.
Yeah, well, bad publicity is not news for the bat ecology research community. We, of course, it's disappointing, and it can be challenging to explain to people the really complex sort of issue around how viruses are transmitted between people and wildlife. And it is frustrating that bats are getting an unfair sort of bulk of that. But bats get bad publicity all the time anyways. So I'm pretty used to being like, well, here's the information that I think you're missing. And in reality, you know, the flying mammals that are really good at transporting diseases is us when we get on airplanes and we travel around the world. So that's sort of how I've been dealing with it, but I haven't had to deal with it too much. A lot of my colleagues have done a great job of getting good information out there for people, and I've just tried to do my best in conversations like this.
Yeah, yes, I totally agree with you. It's like it's more the impact that the human had on like the wildlife has to stay wild, and you shouldn't mess with that. So I totally agree with you. And Francesco is asking one more question. Do you take special precaution when you investigate them? Yeah, good question.
So it's not. I'm not clear to if you mean do I take precautions to protect myself from bats, or if I'm taking precautions to protect bats from us. But the answer is yes and yes.
Both can be.
Yeah. The answer is yes, and the answer is also yes. So when I work with bats right now, I'm rabies vaccinated. I'm always rabies vaccinated. I get my blood checked regularly to make sure I have proper antibodies. So, and I wear hand protection so that even if a bat does bite me, chances are it's not going to break the skin. So I'm protecting myself from that disease. And then also we often wear latex, and we're washing our hands very often, especially between bats. Because now what our main concern is that we're actually really worried that people will give coronavirus to bats.
Yeah.
That's one of our main concerns. So this summer when I'm going out and mist netting, capturing bats that are alive, I'll be wearing a mask. I'll be wearing special clothing that I only wear at a mist net site. We'll be wearing gloves and disinfecting between bats, and disinfecting whenever we touch something that a bat might come into contact with, so that we don't give them our cuties. That's our main concern, is really not getting the bat sick.
Yes. And I have one question. If you have some special anecdotes about the times that you went to look for bats, or like some interesting adventures that happened.
I've got loads. I don't know. I've got loads. I like I said, I feel really lucky that I get to work with these animals hands-on, and I've had opportunities to do it in lots of different kinds of places. I don't have any specific stories off the top of my head, I'm afraid, but I will say that it's an exciting field to be a part of, and I'm always learning, and learning is sometimes messy and often leads to funny stories, I suppose.
Yeah. Yes. Okay, let's see if there is more questions. No, I have one thing. What motivated you to come to Norway to study bats?
Yeah, so I'm actually a Norwegian citizen. My mother's North Norwegian. Yeah, that's part of it. I mean, I also had a bit of a quarter life crisis, to be honest with you. I was sort of needed a big change, and it was a good time for me to go and live in a country that I've been a citizen of my whole life and never lived in before. And also, I knew that there was this opportunity to study bats.
Yeah.
In a master's degree, which overlapped well with where I was in my career. So the stars aligned, and I went for it. And so far, I feel good about that.
Yes, that's great. Thank you so much, Abril.
Yeah, thank you very much. It was fun.
Yes. And now I give back the screen to Juditha for closure.
Yes. And I would like to have all of our speakers. I think Coline had to leave, unfortunately, but all of the rest I would like to have here, and as well our volunteers. And now I would really like to thank you so much, all of our speakers. It was really a great evening, I think, and very interesting talks. We heard of the cold temperatures in Antarctica and fruit fly insects and like singing bats. So this was really amazing, I think. So I would like to give them a big applause and thank them so much for this evening. So it was great having you here. Thank you. And now I would like to share my screen once more. Yeah, because now this is the end of the Pint of Science Festival in 2021, and we hoped you enjoyed all the talks today and in the previous days. And we were happy to have such a great audience as well, besides our speakers, with many interesting questions and thoughts. And I think we all really enjoyed discussing with you. And I want to thank once more our volunteers. And of course, I want to thank as well once more our sponsors who made this evening happening. And now I hope to see you next year again, and then hopefully as well in person in some bar here in Oslo, that we can really enjoy a pint together and have it in real person again. And with this, yes, with this little cute talk and the dates for the next Pint of Science event in 2022, I hand now over to Lourdes and the quiz of today. So I hope you will join there.
Hello, everybody. Welcome to this last session of the Pint of Science Oslo quiz. And after 3 wonderful talks we just heard, we have a simple yet informative and fascinating quiz for you. So let me share my screen first. And give me a second. There you go. So now here in the screen you can see the pin you will need to join our game. And while the people wanting to join us, I will talk a little bit about the quiz. So just feel free to join whenever, and we'll start soon, okay? So, well, here's the deal with the quiz. I hope you all pay attention to the talks, because the information the speaker gave us is going to be very useful here. So what I'm going to be doing is that I'm going to read the questions out loud for you, and the same for the answers. But be aware that the faster you answer, the more points you will get. So you don't need to wait for me to finish reading. Just answer as fast as you can or you know, okay? So as I mentioned at the beginning of the night, the person who wins in the quiz will get, well, first we're going to say congratulations, of course, and later on they will receive a prize. So to get this prize, the winner will need to send us an email at norway@pintofscience.com, and this is shown either in the screen or in the comments, I think. And yeah, I actually think that's all we need to know before we start the game. So we have around 14 people. Welcome. And I hope you're as excited as I am. And I guess here we go. So let's start this. One, go. So what is the maximum depth of ice in Antarctica? So 2010, 3,185, 4,776, or 5,243. And the answer is, oh, wow, you guys did pay attention. The answer is 4,776. And I think that's really seriously thick ice. So let's see. Batman. Batman is on top of the score right now. Let's see what we get after. So question 2, how long can a fruit fly live? So I'm always wondering about this every time I see fruit flies at home. So first we have 10 to 15 days, 10 to 15 hours, 40 to 50 days, or 6 to 7 days? And the answer will be 40 to 50 days. Wow, this is going to be quite tight, I guess. Everybody's guessing. So yes, fruit flies can actually live several months, as you heard in the talk. So if you take good care of them, of course. So to be honest, I really thought they live less. So, I wanted to mention, remember, you can still join the game whenever if you just type the code at the bottom of the page. So let's go for the next one. And badminton is still on top, but it's April is close. So let's continue. So how many bat species are documented in Norway? 5 species, 55, 14, or 12? I think this was in one of the slides. And let's see if we can get the right answer. And the answer is 12. Very well. So yes, there's actually more than around 90 mammals inhabiting Norway, and from those 12 are bats. So it's really not a bad number of species if we consider that, well, it's really cold here in Norway. So next question. And April is coming on top. Let's continue. And how many % of the world's fresh water is stored in Antarctica? 50%, 85%, 15%, or 70%? So I'm wondering how easy it is to get this fresh water. Okay, the answer is 70%. Yes. So yes, indeed, in Antarctica, we can find more than 90% of all the ice in the world. So that's a lot of water. And however, Antarctica is actually considered an arid place. That's really interesting. So let's see how the board is going. And we have green on top, but it's actually really close between green and April. Oh, wow. Let's see. Next question, true or false? Fruit flies can get cancer in the wild. True or false? And the answer is false. Yes, this might be surprising for some people. It was surprising for me, actually. But fruit flies can only get spontaneous neoplastic growths, but no cancer. So let's see. We have green on top for a little bit more now. Let's continue. And which bat species sings from the top of the rooftops to find mates? The northern bat, the party color bat, whiskered bat, or all of the above? It would be pretty cool to have a bat singing on your rooftop, I think. Yes, the party color bat. I think April mentioned it was her favorite bat. Yes, I would really like to see this bat. So this bat is just a song-flighting bat, and as a random fact, if there are many males song-flighting, the air is going to be filled with a zooming similar to that of the cicadas. So yes, that's really nice. Let's see how the board is going. There's a little bit of a change. Now I can also see Snape, Nora coming up. Let's continue with the next question. So, what was the coldest temperature measured in Concordia? Minus 84.6 degrees, minus 196, minus 75.4, or minus 36.3? They all sound pretty cold to me. So the answer was minus 84.6 degrees. Very well done. And yes, this is really one of the coldest places on Earth. And this temperature that we can see here in the screen was recorded, I think, in the winter of 2010. And it was actually one of the coldest temperatures ever recorded on Earth. So let's see how the score is going. And April is back on top, but green is not far away. So next question. So how many chromosomes does a fruit fly have? 8, 4, 12, or 2? You can also think about pairs of chromosomes here. And the answer, yes, it was 8. So 4 pairs of chromosomes. And as it was mentioned in the talk, 1 pair will determine the sex, and the other 3 are autosomal chromosomes. So basically not determining the sex. So let's continue. And Snape is surprising us and coming on top. Good. Let's go next. And what percentage of the Norwegian mammal species are bats? So 32%, 50%, 2%, or perhaps 30%. And the answer is yes, 32%. That's right, guys. So yeah, we know that the elk and the reindeer are the most perhaps common mammals in Norway, but I really think bats are quite abundant too. Perhaps they're just harder to see. So next question. And April is coming back on top again. This is really close. Next question. Again, true or false? So, space medicine is another kind of research in Concordia. True or false? And the answer is true. Yes, indeed. So, Concordia Station has been proposed as one of the real-life Earth-based spaces. So, for to take long-duration space missions. And this is because there are many stressor characteristics similar to the ones that the astronauts will face in space missions. So yes, space medicine. So let's see the next question. And before that, April is still on top, but Snape and Batman are still here. So let's go next. And what is the color of fruit flies in the wild? So we have red eyes and green body, brown eyes and red body, brown eyes and brown body, and red eyes and brown body. So did you guys notice Helen's drawings here in the talk? The answer is red eyes and brown body, yes. So in nature, these tonalities actually vary, of course, and the body is yes brown, but also kind of yellowish brown. And also they will have some black rings across the abdomen. So yes, next time you see a fruit fly, perhaps you can try to see those details. So let's go and see how the board is going. Batman is coming closer to April, and we have Snape, Marmee, green. And let's go next question. So when can you find bats in Oslo? So spring, all year, winter, or perhaps May? And the answer is yes, all year. So yeah, we have plenty of opportunities to see bats. However, it's important to remember that most of the species is easier to see them in the summer, as April mentioned. So pretty soon, actually. And let's keep an eye in the sky to search for bats then. So let's see how the final score is So Marmy, congratulations, you have the 3rd place. And then Snape, the 2nd place. And then the winner is Abril. Congratulations to our 3 players that made the podium. Very well done. Congratulations to everybody, and thank you so much for joining us in the quiz. I hope you guys had as much fun as I did. I am gonna stop sharing my screen. And I just wanted to remind you, please, the winner, don't forget to contact us. The details are, send your details at norway@pintofscience.com. I think it's going to be in comments. So please just send an email with your details. And for all the players, amazing job, guys. You did really well. I'm really, really happy. So with this, I actually say goodbye to you all, but I would like to welcome in the screen the other volunteers of the night, perhaps, if they are around. Yes. So we wanted to say thank you for joining us tonight and for the past 2 days in Pint of Science here in Oslo. We are all really looking forward to see you next year. So I don't know if someone would like to say some other words. But if not, perhaps we can just say then until next time, have a good evening, and thank you so much for joining us. I hope you enjoyed this event. Bye-bye.
Bye.
Bye-bye.